It's my great pleasure to introduce our keynote speaker, Dr. Chris Walsh. Chris Walsh is a Howard Hughes investigator, and he's also the chief in the Division of Genetics and Genomics at Boston Children's Hospital. He is also a professor of pediatrics and neurology at the Harvard Medical School. And as you know, he's a member of National Academy of Science, and he has won many, many awards. And most importantly, he has won the Gruber Prize for Neuroscience, as well as the Cavalier Award in Neuroscience for his coverage of genetic basis for many brain disorders. And for he is the first one to use retrovirus for in vivo lineage tracing in the nervous system. And he's also the first to develop DNA barcode using retroviral strategies for linear tracing in study brain development. So without further ado, let's welcome Chris. Thank you.
For that very generous introduction. Modesty, it was Connie Sepko who developed the retroviruses. I just added the barcodes, but I'm happy to be given credit for that anyway. But I'm thrilled to be able to talk here a little bit, and it's just been a really wonderful meeting. And so I'll just sort of dive right into it. So I'll be talking today about somatic mutations, and actually they occur with every cell division. 2 or 3 or more, 2 or 3 SNVs, other things like transposon insertions and copy number variants. They can occur early, in which case they'll be shared in all of the tissues of the body, but only in a fraction of the cells in those tissues. They can occur a little bit later, after the brain tissue has separated from the non-brain tissue, in which case they'll be restricted only to the brain and probably in a smaller fraction of cells, since they're arising at a time when there's a larger number of cells in the body, or they can be in one hemisphere and not the other, or even in a little patch of the cortex. And so we've been obsessed with these now for the last decade or so, and they seem to play roles in a variety of different conditions. I'm just going to tell you today about 3 short vignettes, and they illustrate, they raise questions about whether somatic mutations in the human brain might behave a little bit the same way somatic mutations act in cancer. So in cancer, we know that the risks from genetics and risks from the environment, like smoking, they interact by creating or amplifying somatic mutations. And I'll raise the possibility that similar things might apply in some brain diseases. I'll talk about activating RAS-RAF-MAP kinase mutations in temporal lobe epilepsy. I'll talk about single nucleotide variants in schizophrenia, and I'll talk about this is actually somatic copy number variants. That S should be a C, but I'll get there when the time comes. So a lot of this work started with someone who's been a former postdoc now for more than a decade, who now actually is the as of October 1st, is the associate director of the NINDS, Ann Paduri. And she wanted to figure out the brain of this young boy named Dante, who basically from the moment he was born started having intractable seizures. And he was weak on the left side because this is his right hemisphere and it's overgrown and dysfunctional. And so in a desperate attempt to control these seizures, surgeons removed his entire right hemisphere, right down to the thalamus. And this is what his post-op picture looked like. There's no brain there, it's just CSF. And this was life-saving to him, remarkably. He had no seizures for 6 years. His seizures remained well-controlled. He learned to walk. He learned to talk. He speaks fluently. He reads at grade level. He's in his teens now. He'll tell you he's completely normal. And I use his picture with his parents' permission. He's still a bit weak on the left side, but basically they salvaged his life. And so Anne wanted to figure out what the heck's going on with a brain like that. And Dante actually had an activating cancer mutation that was restricted just to that one hemisphere. It was an activating mutation in a gene called AKT3. And in the subsequent 10 or 15 years, this is now canon, that patients with these intractable forms of pediatric epilepsy, with these particular abnormalities on MRI scans, have activating mutations invariably in the mTOR signaling pathway. And with the most sensitive signaling methods, you can identify mutations in at least 80 or 90% of these cases in the mTOR pathway. And so these hemispheric lesions called hemimegalencephaly, the mutation is actually not in all of the cells, but only in 10 or 20 or 30% of the cells. And in these much smaller lesions, which are by far more common, and are in fact the most common cause of intractable pediatric epilepsy that results in brain surgery to treat it. The mutation is present in a tiny fraction of cells, maybe 1, 2, 3%, and only in the lesion and undetectable in the blood, and we assume it's undetectable in the brain. And so we just get the part of the brain that they take out to treat the epilepsy, and the epilepsy surgery works very well. Surgery in a well-selected case of a kid that looks like that will be curative in about half of the cases. And then we can study that, and Alyssa DeGamma, a former student, followed up on Ann's work. And so these are stable malformations. They're not cancerous, although they have classic cancer mutations, all up and down the PI3 kinase mTOR pathway. These are mutations first described in cancer, but they are mostly in neurons, which, as we all know, do not divide, and so they just cause other kinds of mischief by making those neurons inherently epileptic. So these kids tend to have seizures starting right away. These kids tend to have seizures starting sometime after birth. So there's a relationship between the frequency of the variant, the fraction of the cells that carry it, and the severity and of the seizures and the disability. But amazingly, you can still get intractable epilepsy from a little patch where the mutation is present in fewer than 1% of the cells. And this has allowed mTOR inhibitors, originally developed for the treatment of cancer, to be repurposed to try to treat kids that don't respond to surgery, although most of these don't penetrate the brain very well, so they haven't been as successful as we might have liked. And there's important work on this from Stephanie Baulak, Bill Dobbins, Joe Gleason, and Jung-Ho Lee in South Korea as well. So how about the most common form of focal epilepsy in adults? That's called mesial temporal lobe epilepsy. Epileptologists, for some reason, like to call the medial temporal lobe the mesial temporal lobe, or MTLE. And so this has been known forever. Hippocrates and the Greeks referred to it as a sacred disease because patients often have psychic auras with emotions or memories or deja vu rather than convulsions. The typical age of onset is not as a child, but as a teenager or as a full adult. And then these psychic auras often lead to speech arrest, and there's only a convulsion if the seizure generalizes beyond the temporal lobe. This accounts for 2/3 of all intractable adult epilepsy, and it's never been fully explained why it happens in the temporal lobe, because the pediatric epilepsy happens all over the whole neocortex. So this mesial temporal lobe epilepsy actually has a pathological hallmark. It's called mesial temporal sclerosis. And this is characterized by neuron loss and glial loss, which you can see radiographically as a little bright spot in the hippocampus. And pathologically, you can see there's the neurons in CA1 and CA2 are completely wiped out. For those of us who think the hippocampus is really important in learning and memory, that's a kind of a scary thought, but it's usually unilateral. And people seem to do generally not too badly despite that. Now, what are the genetics of mesial temporal lobe epilepsy? There really is none. It's the classic complex form of epilepsy. There are no Mendelian forms. Most patients actually report, 2/3 of patients report a non-genetic risk factor, such as a history of head trauma or a severe single seizure as a young child. Inherited genes, whether common variants or rare variants, have so far been shown to have little or no role. And yet it's been known for 75 years, since the days of Wilder-Penfield, that if you take out the temporal lobe, if you can localize it using EEG to the temporal lobe and then remove part or all of that temporal lobe, that again is curative in more than half of the patients and causes at least some improvement in more like 70%. And so we wondered whether there might at least be some mTOR variants in there causing some cases of mesial temporal lobe epilepsy. So Sattar Kushku, an epilepsy fellow who now has his own lab at the Brigham, was brave enough to take a shot at that. And so initially, we analyzed 100 cases, many of these from Yale, from Dennis Spencer and Chris Colley, who conveniently moved to Boston and asked to collaborate with us. And then we had also additional cases from Boston Children's and elsewhere. We did what we thought was pretty deep exome sequencing, 500x coverage, to detect these variants that are only in a fraction of cells. But we found a grand total of only 11 somatic mutations predicted to be damaging in those more than 100 cases. But amazingly, 10 of 11 of them were all in the same pathway, the RAS, but not mTOR. None of them were mTOR, in the RAS-RAF MAP kinase signaling pathway, and we found none in controls. And these were all predicted to be activating. Again, classic cancer mutations in PTPN11 encoding SHP2, the classic RAS G12D and G12V mutations, the classic BRAF V600E, and then loss of function mutations in negative regulators, notably neurofibromatosis 1. So in work that's not published yet, Sattar has collected more than 500 cases from epilepsy surgery centers all over the world, because this is a very common type of epilepsy surgery. And we find that there are more than 100 of these cases have activating mutations in PTPN11 alone. You see all of these things this is 10 cases with this mutations at the same site. Here's another dozen cases with a mutation at that amino acid. So we see these highly recurrent patterns of mutation. There's another 100 cases that have activating mutations in other RAS pathway genes. 3/4 of these cases show nothing but mesial temporal sclerosis and no evidence of tumor. In fact, these mutations are more common in cases without tumor than they are in cases that have an associated tumor. And typically the mutation is, again, present in like 1% or so of the cells. But this deeper sequencing also reveals that some normal people have activating mutations as well, but there's still a strong enrichment of mutations in cases compared to controls. And we're just finalizing and collecting our numbers, so I don't have a figure to illustrate that. And so this has important therapeutic implications, again, because this pathway has obviously been targeted extensively in cancer because it's one of the most important cancer pathways. Now some of these drugs can be repurposed to see if they can be useful for intractable adult epilepsy. So why do the neurons die? This is a degenerative condition where you have this wipeout of CA1 and CA2. We don't know if that's an effect of the mutation or of the seizures or some interaction. What cell types carry the mutation? Sattar's done a little bit of sorting of neurons, oligodendrocytes, and astrocytes, and clearly the mutation is present in the neurons and in the neural glia. So we're fairly confident this is a mutation arising in neural precursor cells as opposed to, for example, microglial cells, which I can talk about on another occasion. And so what we find is that we have RAS MAP kinase mutations predominantly in the temporal lobe, and then mTOR everywhere else in the neocortex. And this enrichment of different pathways in the different regions, it was actually just significant even based on our first paper. And so presumably this reflects the very different biology of these two different cerebral cortical structures, the hippocampus or the archicortex and the neocortex. So there are a couple of other features of these. This is just our first 11 cases, and we find invariably that the mutation is more common in cells of the hippocampus than in the neighboring temporal lobe neocortex. In order to get to the hippocampus, you have to go through the temporal lobe, and so you get this neocortex removed as well. And so sometimes we had tissue from both sources, and when we did, the mutation is always more common in the hippocampus than the neocortex. Here's an example where it's like the variant allele frequency is 3.3, meaning that the fraction of cells carrying the mutation is twice that, where 6.5% of the cells carry the mutation. And then it's barely detectable in the temporal lobe neocortex, or here it's undetectable in the temporal lobe neocortex. The other thing you observe is a similar relationship that we see with mTOR variants, that patients that have mutation in a large fraction of cells, like this 10-year-old male, actually have surgery at a young age because their seizures are rapidly intractable, whereas people who have surgery at late stages, whose epilepsy was presumably later in onset, have the mutation in a smaller fraction of cells. So there's the same dose-response curve in terms of the epilepsy. But what's amazing about these somatic epilepsy variants in the hippocampus is that they are subject to postnatal effects, because I think most people here know that the hippocampus is the one part of the cerebral cortex that continues to form neurons after birth. Okay, specifically the dentate gyrus of the hippocampus. And there's this vast literature, led, of course, by Rusty Gage, among others, but that show that this hippocampal neurogenesis can be modulated by a host of things. Activity, seizures, trauma, drugs, and other things. And so the known risk factors for human temporal lobe epilepsy, namely a seizure as a young child or a history of head trauma, both of them induce a wave of proliferation in the hippocampus. And so this leads us to suggest a model for how life might interact with genetic risk for epilepsy, that basically so RAS activation in neurons is known to be inherently epileptic. There was a lovely paper from Jung-Ho's lab showing that. And so many of us might be born with a small clone of RAS-activated neurons. And so this is a latent risk that might be present in some normal people. We think about 20% of normals have these. But then an environmental risk factor may stimulate a wave of proliferation, and that proliferation is going to favor cells that have oncogenic cancer mutations, either because they proliferate more or because they survive better or both. And so that would lead to a higher fraction of cells in the hippocampus carrying that RAS mutation. And we know, I just told you that increased dose causes the epilepsy to be more severe. So this model can explain, first of all, the predilection of adult epilepsy for the temporal lobe in the first place, and second of all, this phenomenon that clinicians talk about, that seizures beget seizures, that a kid might have his first seizure when he's 16, but then his second seizure may come 6 months later, his third seizure might come 6 weeks after that, his fourth seizure might come 6 days after that. So there's something about epilepsy which is inherently epileptic. So now for another vignette about schizophrenia. So is there a role for somatic mutations in schizophrenia? And this is a paper that was just published last month in Science. We're specifically talking about somatic point mutations. So these are somatic single nucleotide variants, or SNVs. And so this is work of a former student, Eduardo Mori. It was really started by Andy Chess and Attila Jones, a former postdoc in his lab. We had a joint grant from the Brain Somatic Mosaic Network to do this. And Vova Sepelarski, who's a postdoc in Shamil Sanayev's lab, gave us really important help with the bioinformatics that was really led by Eduardo. And so what we wanted to find, what we wanted to test, was whether mutations occurring in neuronal precursor cells might create risk for schizophrenia, in addition to all of the other risk factors that act through the germline. And so to do that, Attila and Andy separated neurons using new end sorting, and then made DNA just from the neurons. And so we could focus on clonally shared variants in the neurons and ignore those that might have occurred later in life in the glial cells. And so then we did whole genome sequencing, actually, at about average of 267x. And so again, this can detect variants that are present in about 2 or 3% of the cells or more. And so we identified, we tested, we had a very, very high validation rate. We find a sort of nominally larger number of variants in schizophrenia compared to control. But this is after actually we removed, one of our schizophrenic cases had like 5 times as many somatic variants as anything else. And that can happen in normal people. So we set that one aside. And then after that, we still saw nominally more variants. But actually, this non-significant p-value is with the most conservative possible statistic. If you use a Poisson test, in fact, this is significant at 10 to the minus 4th, even after removal of that outlier. But we used a permutation test. In fact, we couldn't even get all of our cases to fit in the permutation test. And so we suspect that if we had a larger sample size, which is what we're trying to collect now, there would be a significant overall burden. But what we do see is highly statistically significant burdens of particular types of somatic mutations in schizophrenia compared to controls. And so they're enriched at open chromatin, DNase hypersensitive regions in schizophrenics compared to controls, and in fact are depleted at closed chromatin in cases compared to controls. We specifically see enrichments not just in open chromatin, but specifically very close to transcription factor binding sites. Here you can see the enrichment is within 50 base pairs of the transcription factor binding site, and then rapidly fades away as you get even 100 base pairs away from it. And so we think there's something very specific about the transcription factor binding site that increases the rate of mutation at those sites. And it's not just any transcription factor binding sites. The ones with the highest nominal enrichment are transcription factors where the transcription factors are bound in the brain, either in neurons or in neural precursor cells, although we see enrichment for other developmental patterns as well. And so we think that this reflects that these mutations occur during development, but notably in the development of the brain. In fact, the individual transcription factor sites with the highest nominal enrichments are for transcription factors that have essential roles in head and brain development, things like AP1, SP1, and retinoic acid receptor. And beyond that, we saw particular patterns of nucleotide substitution. So mutagenic forces don't just create random mutations, they create specific types of mutations. So ultraviolet light changes C's to T's, or cigarette smoke changes C's to A's by oxidation of the guanine. And so what we found, we found two patterns of enrichment in schizophrenia. They showed a 20-fold enrichment of CpG transversions specifically at transcription factor binding sites, within 100 base pairs of transcription factor binding sites. And looking back on a previous study we'd done on autism, we saw an insignificant trend towards a similar kind of enrichment for these CpG transversions. This is a change of a C to a G or an A. And interestingly, Vova had previously described what causes these CpG transversions. So in cancer, this is due to a very specific type of mutation. They occur basically when there is an unrepaired A basic site and it gets replicated before it can be repaired. And so that causes these particular patterns of CpG. And in fact, they're thought to arise on the basis of fairly good evidence during the first few cell divisions, when we have a methylated genome coming from the sperm and a methylated genome coming from the egg, and they create a zygote that requires a completely different pattern of methylation. And so the genomes of both parents become globally demethylated, and demethylcytosines are very vulnerable to oxidative damage, and then the genome becomes remethylated, but in a different pattern, consistent for the zygote. And so that would predict that these mutations would occur in the very first few cell divisions and be, in an eighth or something like that of the cells. And then we found a second pattern of enrichment, which again was very specific. These are T to G mutations enriched on nominally more than 50-fold. I mean, you never see these kinds of enrichments. And again, a highly statistically significant one. In fact, overall, the schizophrenic brains showed larger burden of T to G mutations than controls did, again, with a very significant p-value. But the most amazing thing is that we found three pairs of unrelated schizophrenic individuals who had the exact same mutation in the exact same location in the genome. So that these two cases, one from Mount Sinai, one from Pittsburgh, showed the exact same variant at the exact same location on chromosome 21. And then these two cases had the exact same variant as each other in a different site on chromosome 21. These two had, again, the exact same variant at yet a third case. This is absolutely ridiculous. This is like this sort of thing. The chance of this happening is like 1 in 10 to the minus 10th. This is impossible that this could have happened by chance. And what this seems to reflect is that it's not the whole genome that's being mutated. I mean, it might be the whole genome that's being mutated, but it's not the whole genome that's being stably mutated in a double-stranded mutation. But instead, somehow this transcription factor binding is channeling these mutations to not the whole genome, but to a very, very tiny fraction of the genome, to just transcription factor binding sites and just those that are active during development. So, as I say, Vova is an expert in cancer genomics, and by the way, he is on the job market this year. And so we wanted to figure out what is this particular pattern of T to G substitution. So again, we turn to the cancer literature. And the cancer literature, there are now more than 100 different recurrent patterns of nucleotide substitution in cancer. And this just shows about half of them. And so the particular pattern that we found to be enriched in schizophrenia is called number 17B, or single base substitution pattern 17B, which is characterized by these T to G changes. And so we looked back to ask, does this happen in cancer too, that you get this amazing recurrence of the exact same variant at the same location, which had not been described before in cancer, but we found it is indeed the case, that if you look at cancers that contain this signature, we found liver and bladder cancers that had this signature. When you look right at the transcription factor binding site, there is a 100 times increased rate that you'll have the exact same mutation at the exact same site. So this is actually a characteristic feature of all of the 100 different patterns of nucleotide substitution in cancer. This is the only known signature that has this particular feature of this incredible enrichment at transcription factor binding sites that can give two unrelated cancers or two unrelated people the exact same mutation at the exact same site in the genome. And what is it thought to be caused by? It's been explored in cancer in the setting of mutations that occur at cohesin binding sites, another DNA binding protein, where you see there's this enrichment of these sort of ochre-colored T to G mutations, specifically at the cohesin binding site. And it's was shown in this paper that this seems to relate to the fact that damage happens all over to the genome, but then the DNA repair apparatus rushes in to fix it, but it can't rush in to fix it at transcription factor binding sites because the proteins bound to the DNA are in the way of the DNA repair apparatus. And so those mutations are slower to be repaired, and so hence can be replicated before they can be repaired. Then what causes them in the first place? Nobody really knows, even in the setting of cancer, what causes this signature, but the best bet is that these are they're thought to reflect incorporation of oxidized, specifically oxoguanine, into cells that are rapidly replicating, and then incorporated oxoguanine can form a false base pair, and so that can ultimately result in either an A to a C or a T to G on the opposite strand. These two substitutions represent the same mutation, just on opposite Watson and Crick strands. And so we think that the transcription factor binding then, by occluding DNA repair, channels these to a tiny fraction and causes this amazing recurrence. Many of these T-G mutations change regulatory function. This is lovely work from Tan-Tan Nguyen, who was in Kristen Brennan and Steve Riley's lab at Yale, where they tested all of these variants in MIPRA assays. There are several of there that affect dosage-sensitive genes essential for normal brain development. One is in a promoter for ELAV3, another is a promoter for NFIX. This is particularly intriguing because heterozygous loss of function in humans causes brain overgrowth because this is a negative regulator of stem cells, and it causes a behavioral feature with prominent behavioral features called the Malin syndrome. But the T to G ones are most likely to alter, and it's no surprise that these mutations that are occurring right at transcription factor binding sites would alter the enhancer activity of the sequence. So we think that these somatic CPGs and these T to G mutations are two different processes because they also occur in different cases, and they also occur at different variant allele frequencies. So the CPG mutations are present at a very high variant allele frequency, 0.18 on average, which means they're present in about a third of the cells, and that's very consistent with the idea that they occur at the 2 or the 4 or the 8 cell stage. And then the T to G mutations are present in a smaller fraction of cells, suggesting that they don't start right away, but they start a little bit later, but probably also in the first trimester to get this sort of relatively large variant allele frequency. And then we see that some cases have one and some cases have the other. Most cases have neither. And so of our 62 schizophrenia cases, you see that the CPG transversions are found in 3 cases. The T to Gs are found in 5 cases. Again, we removed this outlier for all of the statistics that I showed you. I'm just putting it back in for this one summary thing here. So we think that these are 2 different processes. The CPG, we think we understand what it is. The T to G, we still don't really understand what it is and would love to find out. So we don't really know that these contribute to risk. We think they probably do because they're enriched in cases compared to controls, at least particular types, and because they can be damaging. But it's possible that something about the schizophrenic brain either permits these mutations or blocks their repair. And so we would like to know what prenatal we think that the T to G reflects some sort of prenatal mutagenic mechanism, and we don't know what that might be, potentially relating to oxidative damage, given what we know about the signature, And one likely or one suspicious candidate is maternal infection or immune activation, which has been known for decades based on epidemiological studies to be a risk factor for many different psychiatric disorders. But the point I want to leave you with is that whatever that process is, it's likely to be a nonspecific process. But even a nonspecific process can cause very specific mutations because of this process of channeling of the mutations by transcription factor binding sites. So finally, I'll talk a little bit about how do somatic mutations in neurons evolve during normal early development. And so cells with abnormal genomes, like losing a whole chromosome, are amazingly common during early embryonic development. This is a recent paper from Chow's lab where they looked at blastocysts. You see gains and losses of chromosomes in about a quarter of the cells. This has long been known in the IVF literature that they fertilize and then they'll check a karyotype and they get really scared because they see a lot of aneuploidy, but surprisingly, many of these aneuploid embryos, if they actually are used, they seem to do just fine. Even at post-implantation studies, they still saw a large rate of aneuploidy. Even in cortical neurons at early stages, they still saw maybe like 5 or 10% of the cells were grossly aneuploid. Now, that can't possibly be good for neurons. We know that neurons are very sensitive to gene dosage. Copy number variants in the germline are associated with various kinds of epilepsy, but more importantly, loss of a single allele of about 1,000 different genes is associated with epilepsy, so that we know that neurons really do want to have 2 copies of all of their genome. So Diane Shao, a really smart pediatric neurologist, MD, PhD, who's also actually on the job market, wanted to see what happens to these copy number variants over the course of development. She worked with Yifan Zhao, a graduate student in Peter Park's lab. And so they developed a new method to analyze them in a single-cell way by using TAGMENT TN5-based amplification. And then developing a new algorithm to analyze them at large scale. So basically, this is what copy number variants look like. They analyzed neurons from the cortical plate, so specifically focusing on postmitotic, non-cycling cells. And when you do RNA-seq on those, you see most of them are excitatory neurons and some are inhibitory neurons. And so this is what it looks like. Basically, the chromosomes are arrayed across from 1 to 22. And so these are cells that have very aberrant genomes that have gains or losses. This is a cell that looks pretty good at diploid at copy number 2. But this kind of CNV calling from single cells can be very artifact prone, but their method, just by, you might imagine you just do it by counting the number of reads from a sequenced genome of a single cell, but that can be complicated by amplification bias. And also the amplification destroys the ability to validate what you're seeing. And so they developed this new method. What happens is that the TN5 inserts at particular sites, and so those insertion sites are different on the maternal allele and the paternal allele. And so if they're a copy neutral, you'll see 2 sets of insertion sites, one for each of the 2 chromosomes. But if there's a copy number loss, you'll only see 1 set of insertion sites. And if there's a copy number gain, you'll see 3 sets of insertion sites. And so you can use the insertion sites in addition to the copy number as 2 independent means to analyze copy number. And so this is what they found in prenatal neurons, and this is what they saw in postnatal neurons. And so you can see there's a lot going on here. The gains are in green, and losses are in red. And you can see that a large, and again, the neurons, the chromosomes are arrayed in a circle. Here's chromosome 1, 2, 3. So from largest chromosome to smallest chromosome. And so just with your eye, perhaps, you can see there's a lot more color in the prenatal neurons than the postnatal neurons. And now copy number variants also are categorized by the cancer people into literally 90 different recurrent patterns of copy number change. There are only 4 that are relevant for analyzing neurons. And 1 is essentially diploid with a subchromosomal gain or a loss somewhere. 2, it got a lot more going on. You'll see that the third type, CN1 and CN9, has a lot of focal losses. And then the most abnormal is the CN1, CN13, which has broad losses in a lot of different sites in the genome. And so what you see is that the prenatal neurons have larger numbers of copy number variants, and then the postnatal neurons are more likely to be green, more likely to be mostly diploid. And so somewhere near the end of gestation, there is somehow a purging of neurons that contain large-scale copy number variants. So what causes the copy number variance? The copy number breakpoints are correlated with replication timing. So that suggests that they arise just as I suggested that they do, in rapidly dividing cells, and that they occur as a part of DNA replication, consistent with them arising similar to what was described in this prior study. Now, Diane went further, and she looked at a phenomenon which has been, again, described in cancer called micronuclei, because naked DNA, if it gets missed during mitosis, it ends up in the cytoplasm, and that's very toxic to cells. And so it's encapsulated by a small nucleus called a micronucleus. And these, it turns out, are abundant in the developing cortical plate. And then she actually fractionated the micronuclei. You can actually see what chromosomes are overrepresented in the micronuclei. As you'd expect, the larger chromosomes are overrepresented, but surprisingly, the smaller chromosomes are overrepresented, 13, 18, 21, X and Y. These are actually the chromosomes that actually are stable as aneuploidies, even in the germline. So what she found is that these large-scale CNVs and the micronuclei dramatically decrease right around the time of birth. So that prenatally, almost a third of the cells have these large-scale CNVs, and then postnatally, it's fewer than 20%. So it's about a 50% drop-off somewhere right around the time of birth. She also found that these micronuclei become much less common postnatally in infants, and actually become seemingly progressively less common as people age beyond the neonatal period. But then there seems to be two phases of elimination of these abnormal nuclei. It's the large-scale changes, which are dramatically eliminated around the time of birth, but then small-scale changes, these subchromosomal CNVs, look like they continue to be eliminated as people grow, so that they seem to be surprisingly less common in elderly individuals than they are in younger individuals, and that actually replicates a finding reported by Mike McConnell 5 years ago, and that there seems to be some ongoing purging of neurons with abnormal genomes throughout life. Now, Eduardo also did a paper with Javier and Alice Lee where they looked at clonal somatic variants. These are single nucleotide variants using bulk RNA-seq from the GTEx and from the BrainVar collection, and found, again, something that was really puzzling at the time. This has very low sensitivity to detect variants. So you only detect a couple in any given brain, but they analyzed more than 1,000 brains, and what they found was, again, exonic single nucleotide variants identified in this bulk RNA-seq were present in more than half of the prenatal brains, and then actually went down by more than 50%. So they were present in fewer than a quarter of the postnatal brains. In fact, the proportion of samples that had a variant that was likely to be pathogenic was, again, much higher prenatally and in the early postnatal period than it was later in life. And so perhaps both copy number variants as well as single nucleotide variants might show this similar pattern of essentially being purged to a certain extent through life. So what drives this process? Well, going back to the copy number variants, we can learn a little bit about what might drive it by seeing where they happen. And so this echoes the sort of survivorship analysis that Abraham Wald did in World War II, where he analyzed where the bullet holes happened in the planes that successfully returned to base after a bombing mission. And so he found that there was lots of bullet holes in the wings and in the back, and that seemed to be consistent with survival of the plane, but that there were never bullet holes in the cockpit or in the engines. And presumably that's because that would kill the plane and the pilot would never return. And so these would be cold spots, places where again, mischief never happened. And so we asked, what are the cold spots in the genome and what do they contain? Those cold spots defined as the regions which are not subject to this somatic copy number variation. So as expected, we found that the cold spots tended to have a lot more genes in them than the hot spots, or the places that were neither cold nor hot. And in fact, the cold spots were specifically enriched for autism-associated genes, which we know to be essential for synaptic development in many cases. And in fact, they're not so much enriched for epilepsy genes, which are often channel genes. The epilepsy genes showed neither any particular enrichment in cold spots nor a depletion in cold spots, but were just sort of average. Whereas, again, the autism genes were strongly enriched in the cold spots and strongly depleted from the hot spots. And so we think there are two phases of elimination of these aberrant genomes. There is a dramatic elimination around the time of birth of these very severely abnormal genomes, and that looks suspiciously similar to the time course of what's been described as programmed cell death, first described in the cerebral cortex by Barbara Finley, where neurons in the cortex just die. And then there is this second phase where there is a more gradual elimination of copy number variants that are less severely damaging, and perhaps point mutations as well, that extends after birth and for a long time period. And that suspiciously overlaps the period that we describe as involving synaptic competition and synaptic pruning. And so it suggests that these processes that are so familiar to developmental neuroscientists, of programmed cell death and synaptic pruning, are actually not just mechanisms of synapse quality control, but are actually mechanisms of genome quality control, that this is how the nervous system eliminates defective genomes. But it also suggests that anything that disrupts that process could leave the brain with a lot of noise, with a large number, as many as a quarter or a half of the neurons, with functionally very abnormal genomes. And so you can imagine that neonatal hypoxia or injury might disrupt this elimination of the most severe genomes, perhaps premature birth. But the most intriguing possibility is that the germline genome might regulate this process of elimination of aberrant genomes. And so we only have a little bit of a hint of preliminary data on that. We happen to have sitting around the lab multi-omic data from 2 age-matched controls and 2 patients that had a germline copy number variant that is strongly associated with autism, the 15q duplication. And so Yifan called copy number variants on them, and the red refers to the most severely abnormal genomes, as you can see from the colors. And you can see that these most severely abnormal genomes are more common in both of the duplication cases than they are in the control cases. But again, this is just a sample of 2. But at least raises the possibility that this might be another way in which germline autism risk genes might act, namely by preventing the elimination of these dysfunctional genomes. So to summarize this third part of the talk, these prenatal neurons frequently have severely abnormal genomes. There's a substantial reduction of severely abnormal genomes. About half of them go away at the time of birth, and then a more gradual postnatal elimination of genomes with more modest abnormalities. That genes essential for synaptic development are underrepresented in the copy number variants of surviving neurons, suggesting potentially roles of synaptic development in regulating that. So just to summarize the whole talk, as my time runs out, these 3 vignettes suggest that, in fact, genetics and environment could interact in the brain in ways that we really hadn't thought as much about before, and perhaps they might give you a new way of looking at some of your organoids. So these activating RAS-RAF-MAP kinase mutations in adult temporal lobe epilepsy, at least they form a possible mechanism that remains to be tested for how life events might influence epilepsy risk by regulating the clonal competition between cancer mutation-carrying cells and non-cancer-carrying cells. And then this mosaic single nucleotide variants suggest ways in which some sort of non-specific process can create a specific disease. Like, why does maternal infection create risk for schizophrenia and not for asthma or inflammatory bowel disease? And it might act by channeling mutations to specific sites in the genome, even specific base pairs in the genome. And then finally, there's this perinatal reduction that suggests that maybe this is one way that germline autism genes and perhaps neonatal exposures might interact to regulate this process of elimination of abnormal genomes. Might also suggest why so many of these somatic mutations associated with disease are these oncogenic mutations, because that might cause them to evade this process of mutation elimination. And so I recognize Satar Elan Wang was a bioinformatician in the temporal lobe epilepsy work. And again, Chris Kale was our principal collaborator. The schizophrenia S&V work, I mentioned Eduardo and Vova, and Attila and Andy. And the fetal work, Diane Shao and Yifan Zhao. And again, that was work that was really importantly done with Peter Park. So thanks very much for your attention.
Okay, we have time for questions. Trainees first.
Hi, Rachel Boyd from Johns Hopkins. I'm right here.
Thank you.
I was wondering if you have any plans to stratify your data by autism severity, if there is any kind of, clearing that is associated with higher or lower functioning autism spectrum patients.
Yeah, well, to explore the interaction of the germline genome with this synapse elimination, there's like a lot of different directions to think about, and that would certainly be one of them. I think for now, we just want to look at the most strongly penetrant forms of autism, like the 15q or 16p, things where we of course, we're limited to studying postmortem brains, and so it kind of depends on what comes our way. But that also, by the way, I don't want to emphasize, the importance of the Autism BrainNet that the Simons Foundation is so generously supported. And so it depends on what's available in that collection. 15q happens to be well represented. Again, the chromosome anomalies are more often diagnosed prenatally, and so they're sort of where we're starting. But then, if there indeed is an effect, if we're able to confirm that there is such an effect, then yeah, it would be great to study all sorts of different things, more mildly acting genes, kids without a recognizable germline variant, to see if there's some range of effects. But again, it's likely to be noisy because the somatic mutations just create a lot of noise, and then there might be differences in how they're filtered. Yeah.
So we have a question here, and then at the back.
Hi, I'm Karina Meyer from the University of Texas at San Antonio. And I thought it was interesting that the neural stem cells, or the proliferative cells, end up being the ones that are, I guess, more likely to get these mutations. Specifically, I wanted to hear your thoughts on the neural stem cell niche. In general, it's known that hypoxia is supposed to kind of, have like a productive environment for them to maintain stem cell-like stemness. Mm-hmm. So I wanted to hear your thoughts about how hypoxia could also be driving both mutations and also being important for the niche.
Well, yeah, I'm probably not the smartest person to answer that particular question. We're very interested in looking at what the effects of hypoxia might be on the genome. But again, that's typically happening at a later stage. And so it will be happening in much smaller clones, and will require really sensitive technology to be able to detect those, those very, very variable clones and things like that. But there's all these weird things about how the glial cells are kind of permanently influenced by neonatal hypoxia. I'd love to find out if they accumulate some pattern of mutations. And again, this idea of this enrichment of transcription factor binding sites just allows for how various non-specific things could cause the same thing over and over again.
Thank you.
Yeah. Oxidational timing, that can generate 5-hydroxyuracil, and that is prone for misincorporation of guanine. So that is a potential explanation of your T2G. That
sounds like a plausible explanation to me. I'd have to sit down and draw pictures of that and the base pairing and all that stuff. It sounds like you might know more about that than I do. So
should actually look at the sequences, if those are kind of sequences which has a higher tendency to be oxidized as well in presence of transcription factor.
Huh. Well, that may also that oxidation, may also play into this enrichment at open chromatin, since open chromatin is again more vulnerable.
There are also reports on that the oxidative demethylation can make a local environment for ROS production that could facilitate a kind of active oxidation, and in your case, it seems that I don't think it's an active process, but it seems to be very prone to certain sequences.
Yeah. Yeah, that's kind of how we're thinking about it, that the reactive oxygen might ultimately generate the oxidized deoxyribonucleotides that get incorporated into the DNA and ultimately give rise to the T to G changes.
But also the transcription factor and demethylation of lysines on histones, by example.
Right.
One of the byproducts is ROS.
Oh, fascinating. Thanks.
Right here, so I wanted you to clarify something that's kind of interesting. You said that in developmental disorders such as autism, you may have a faulty elimination of the damaged cells because there is decreased pruning, right? Which has been actually shown by at least one paper that I remember a long time ago. So, but then it almost means that in that case, the enrichment or the abundance or overabundance of the somatic mutation in these disorders would be caused by a secondary process, which is the default in pruning. And so then it raises the question of what that defaulted pruning would be caused from. What would be the origin of that?
Sorry, the way I was thinking about it, maybe this is what you're saying. Yeah, maybe I was just thinking of it kind of along the lines of, let's say you had a SHANK3 mutation or a Nerexin mutation. And so basically that would be expected to create a defect in this whole process of synaptic competition. Right, and I think it's fair to say some of these germline synaptic mutations cause an observable defect of synaptic pruning. Right, they cause projections to be left, left untrimmed and less specific, and so that they would be acting in this process of synaptic competition.
Okay,
and so that whole process doesn't work properly.
So you're talking about rare mutation. I'm talking about a paper that a while ago, I believe it was from Columbia University, that showed that in garden variety autism post-mortem they saw less pruning.
Yeah, exactly right. And also the Purpura and
those were
Huttenlocher.
just autism cases.
Yeah.
And so in that case, if you have less pruning, then you have less elimination of somatic mutation, but the elimination is a secondary process. Then you wonder why there is less pruning. I suppose there should be another mutation that is responsible for that.
Well, I'm just saying that the synaptic development process may be essential for the damaged neurons to lose out and die.
Last question.
Sure.
Okay, so very interesting. So I have a question about the mechanism of how the SNB is introduced in the brain. So it looks like, so if the ApoBec is involved, so it's a C2U, C2T exchange, but other type are present. So looks like there are ApoBec dependent or independent mechanism. What kind of mechanisms are there, and are there any preferential sites that occur within the brain?
For the CPG transversions or the T to G changes? Well, so neither of them resemble the signatures that have been associated with ApoBec mutations. That has, again, ApoBec has a couple of very specific recurrent patterns of mutation that are other signatures on that list of 90 different possible single base. So we don't have any reason to think that there's a relationship between of these mutations to ApoBec.
Yeah, let's thank Chris again.